Hey guys, today I'm going to take you through one of the most fascinating processes in human embryology, gastrulation. This is a critical stage of development that every medical student needs to master. So, let's dive right in. First things first, gastrulation typically starts around the third week of embryonic development. It's the process where the single-layered blastula reorganizes into a trilaminar structure known as the gastrula. Essentially, we're going from a single layer to three distinct layers: the ectoderm, mesoderm, and endoderm. Now let's talk about the primitive streak. This is a crucial aspect of gastrulation. It appears on the dorsal surface of the epiblast and defines the body's major axes. Think of it as a roadmap for cell migration. Cells move inward at the primitive streak, forming the mesoderm and endoderm. The remaining epiblast cells become the ectoderm. Easy to remember, right? Next, we have the formation of the notochord. The notochord is a rod-like structure that serves as the primary signaling center for the developing embryo. It plays a key role in the development of the neural tube and the vertebral column. Speaking of the neural tube, this is where things get even more interesting. The notochord induces the overlying ectoderm to form the neural plate, which then folds to become the neural tube. This tube eventually develops into the central nervous system. So, in essence, gastrulation is laying down the foundation for the entire body. One important concept to remember is that cell differentiation and migration during gastrulation are highly coordinated. Any disruptions in this process can lead to severe developmental abnormalities. So, understanding the step-by-step development during gastrulation is crucial for diagnosing and managing congenital conditions. Alright, that's a wrap on gastrulation. I hope this breakdown helps you grasp this essential process in human development. Keep reviewing and stay curious. You've got this. I'll see you in the next video.